Greetings of the day, good morning, good afternoon, and good evening. Welcome to the Sartorius Webinar. We are the co-host for today's webinar. I am Ankit Kankar and I'm the Product Manager for BioSpectrum Asia. BioSpectrum is an integrated B2B media platform for the bioscience industry in the Asia-Pacific region. We engage our readers from pharma, biotech, and medtech industry segments through our media products and services at daily, fortnightly, and monthly frequencies across the countries in Asia, US, and Europe. This webinar highlights on antibody-drug conjugates, ADCs, are increasingly being designed and used as highly targeted cancer therapies, a modern magic bullet approach. So before we start the webinar, there are a few house rules which I would like to take you through. All participants are on listen mode only. In case you have any questions, please raise them once. Question and answers window pop up on the screen. We will answer a few questions and answers if time permits. This webinar will be available on demand on biospectrumasia.com for next 1 year. And today's speaker is Ms. Rachel Forfar. Let me introduce Rachel. Rachel Forfar gained a first-class honors degree in biochemistry from the University of Warwick, having completed an industrial placement at Pfizer Pharmaceutical, evaluating the pharmacology of GPCRS in their STS department. Rachel completed her PhD exploring the expressions of GPCRS in yeast at the University of Warwick and went on to a 3-year postdoctoral MRC career development fellowship in Edinburgh. Before joining LifeArc, Rachel is a member of the asset delivery team who collaborates with academic researchers to translate research into lead-stage therapeutics assets by developing and validating innovative cell-based assays. She has experience in a wide range of in vitro pharmacology, cell biology, and molecular biology techniques applied to both small molecules and biologic drug discovery. So we present Rachel. Hi, Rachel.
Hello.
Yes, go ahead.
Right, so yeah, today I'm going to tell you about how we've developed an assay platform for screening and characterization of antibody-drug conjugates here at LifeArc. But before I get into the science, I just wanted to introduce LifeArc shortly. So we're a medical research charity that have over 25 years experience in helping scientists and organizations turn their research into effective diagnostics and treatments. So we form this arc or bridge between the early stage drug discovery and moving this forward, de-risking the targets to become preclinical candidates. And our mission is to pioneer new ways to turn this great science into greater patient impact. So we've got a focus in 4 therapeutic areas. We focus on neuroscience, on personal oncology, infection, and then opportunity-led. So we form this bridge between academic and non-profit institutions and the pharmaceutical and biotechnology markets. So we're from an MRC heritage with a tech transfer company for MRC originally, which is a government-funded research organisation in the United Kingdom. We were established in 2000 and we're for not-for-profit, so all of the money that we make from any partnering events we make is then pushed back into the research. We've got over 180 employees and we've got more than 25 million R&D spend. So we're formed of 3 different arms. We've got the technology transfer arm, we've got the drug discovery arm, which I'm part of, and also there's the diagnostics development up in Edinburgh. So we've recently set up some funds, so if you want to look at any of these on our website, where we can help fund some of the research that's coming through. And also we've got 4 drugs that are marketed, and this is how we've got our revenue stream. So we've got Entyvio, Roactemra, Tysabri, and the main one, Keytruda, which we've got licensing deals from these. So all of the revenues that we've made from these partnering deals have then been ploughed back into the research so then we can fulfill our charitable objectives. So I work in the Centre for Therapeutics Discovery, and this is part of the research team that's found in Stevenage in the United Kingdom. And we bridge this gap between the basic research and early drug discovery So we're involved in target validation, feasibility, generating hits and generating leads. So we work with industry, charities and universities who have these early stage projects, and then we'll take these forward, going through each of these steps, and then try and partner them with either the pharmaceutical industry or biotechnology. And we work in this purpose-built LifeArc facility in Stevenage, which was opened in 2016. We've got about 90 scientists who work here, and we've got state-of-the-art equipment and capabilities. We've got different teams that are able to work together. So we've got biologists, chemistry, and biotherapeutics. So we're uniquely placed to offer this flexibility and be able to work in close collaboration with one another. So we're involved in a lot of different activities in CTD. We've got biophysics capability. We've got different compound libraries that you can ask to get part of them if you're looking for screening campaigns. We've got structural biology capabilities. The medicinal chemistry is involved in rational drug design with the computational chemists. We've got ADME-tox profiling. We've got pharmacology capabilities. We're able to look at high throughput screening of the molecules to give these pharmacological outputs. We're highly involved in assay development as well, with all the different robotics capabilities. And we've also got a strong biotherapeutics team, and our heritage is involved in humanizing antibody. So across the years that we've been working, we used to have this heritage in antibody humanization, but over the years we've branched out and become more involved with antibodies and small molecules, and more recently we've looked at bispecific antibodies, peptide approaches, PROTACs, fusion proteins and peptibodies, but the focus that I'm going to look at today is antibody-drug conjugates. So these, as you're probably aware, have got three main components. We've got the monoclonal antibody that's conjugated to a toxic payload or drug via a linker. And the antibody component is used as the guided missile, which is providing the specificity for the target. And this delivers a toxic payload to the cancer cell. So the advantage of antibody-drug conjugates is that they've got a long half-life and weekly dosing is possible. They're targeted binding to the tumours to give this magic bullet, and they result in high potency. Because they're targeted, they're active only when bound to the target, so they should give low side effects. And because of this, you're then able to use highly toxic small drugs, which wouldn't be able to be used on their own. There is some reports of tissue penetration as well for solid tumours. So a good ADC target is clearly defined as being overexpressed on the tumour and then low or absent expression in normal tissue, so that you get this good therapeutic window. They also need to show small drugs, which wouldn't be able to be used on their own. There is some reports of tissue penetration as well for solid tumours. So a good ADC target is clearly defined as being overexpressed on the tumour and then low or absent expression in normal tissue, so that you get this good therapeutic window. They also need to show good internalisation. So how they work, they bind to the tumour-specific antigen with the antibody part. This is then internalised into the cell and moves through the endosomes to the lysosomes, and here we get proteases and hydrolases which digest the antibody in the linker and release the free drug. This is then able to move into the cytoplasm of the cell and then binds to its molecular target, often microtubule inhibitors or DNA intercalators, and this leads to apoptosis of the cells, cancer cells. You can also get the toxin released into the neighbouring cells, and you get this bystander effect where they're able to take up the toxin, and this causes cytotoxicity in neighbouring cells as well. So I'm going to talk to you about an example we've worked on at LifeArc based on a neuroblastoma target we were looking at. So this is a rare paediatric cancer, and there's only about 100 cases per year in the UK, but it's a cancer of specialised nerve cells called neurocrest cells. And these cells are involved in the development of the nervous system and other tissues. And what happens is in neuroblastoma, it most commonly occurs in the adrenal glands or the nerve tissues of the main body when these neural crest cells fail to differentiate properly, and instead of becoming these mature ganglia, then become neuroblastoma and form these tumours either on the adrenal glands or around the periphery of the nervous system. You get low or intermediate risk neuroblastoma where the 5-year survival rate is pretty high, with more than 95%, but high-risk neuroblastoma, which involves MYC gene amplification normally, is more prone to metastasis, and this is why the 5-year survival rate is down to about 50%. So there's a strong unmet medical need for this disease. So common therapies for neuroblastoma involve surgery, chemotherapy, or radiotherapy, pretty standard for cancer. There have been some immunotherapy options as well with this anti-GD2 antibody. It binds dinutuximab binds to this glycolipid ganglioside, which is expressed on the neuroblastoma cells on the surface. And these antibody dinutuximab is able to bind through antibody dependent cell cytotoxicity, or ADCC, through the Fc portion of the antibody. It's able to cause cell cytotoxicity. And it's approved by the FDA in combination with GM-CSF and IL-2 or retinoids for high-risk neuroblastoma. However, there are problems with severe side effects. So neuropathic pain is one of the main side effects, and this needs morphine treatment to be able to overcome this. And more problematic is cytokine release syndrome, which can be controlled with antihistamines or anti-inflammatory drugs, but this is quite a worry. So there's a need for new therapies and targets for neuroblastoma. So we found that anaplastic lymphoma kinase fulfilled this category. It's a receptor tyrosine kinase. It's an unclear physiological function, but has a role in neuronal development. It's also a known oncogene, and it's got constitutively active mutants in the kinase part intracellularly, as shown here. We've got 3 different mutations with the relative abundance found. And they also get translocations. This is probably where you've more normally heard of anaplastic lymphoma kinase. So it forms translocations of the cytoplasmic part with NPM and ELM4 to make a transmembrane portion, and this is more often found in lung cancer. So ALK has got a pathological role in neuroblastoma, lymphoma, and epithelial cancers such as non-small cell lung carcinoma, colon and breast carcinomas. And it causes increased cell proliferation, so uncontrolled proliferation of the cancer cells. So there are some therapies already in the clinic that target ALK, for example, crizotinib, made by Pfizer, and this is used in the clinic for non-small cell lung carcinoma. And this targets the kinase domain as a small molecule therapy. There's now 2nd and 3rd generations of these tyrosine kinase inhibitors as well. However, the resistance to these is rapid because some of them are resistant to different ALK mutations, or over time ALK forms mutations to become resistant to these therapies. And there's also compensation from other signaling pathways. So we thought it'd be better to use antibodies towards ALK to target the extracellular domain and therefore become a therapy for neuroblastoma. So the rationale of making an antibody-drug conjugate is that ALK protein is upregulated in neuroblastoma. So there's a correlation between the ALK expression and the level of disease stage in neuroblastoma. So as you can see, there's some staining here from patient samples where you can see the vast majority showed staining with ALK antibody to show expression. And there were over 19 that showed strong staining, so high ALK expression. 17 out of the 126 didn't show staining, but the vast majority of the patient samples for neuroblastoma did show this ALK overexpression. And as you can see here, the different risk stage correlates. So the low risk shows there's not many overexpressing ALK. As you get more intermediate, you see more, and then with the high risk ALK, you see a vast majority express ALK. It also correlates with this MYC gene expression, and then you get higher risk and later stage neuroblastoma, and the chance of relapse is much increased. And also there's immunohistochemistry reports that there's low expression in normal tissue. So as you can see by the human protein atlas analysis here, there's only protein and RNA expression really in the brain. So it's restricted to the CNS in adults. So therefore we thought that ALK made a specific tumour marker in the periphery that could be useful for antibody-drug conjugate therapy. So there are some ALK antibodies already in the literature, and these show surface expression of full-length ALK in patient samples. So here in this paper you can see in red there's the ALK expression on the surface here, and this is co-localized with the cadherin, which is a cell surface marker, and when you merge it you see the nice surface expression of ALK, which is going to be important for antibody therapeutic approach. And again, most of these samples were showing ALK expression on the surface. There has been a previous paper as well where ALK antibodies are shown to be internalized, but this was in a CHO overexpressing cell line. You can see here this antibody forms a nice ring of expression on the surface, and then over time you can see that it's internalized into the cells, and you can see these green punctate dots, and this correlates with transferrin in a different color, which is known to internalize. And then you can get over time this antibody then moves back to the cell surface, but we can see here that there is previous examples of the antibodies being able to internalize. So we went about generating some new anti-ALK antibodies, and used a collaboration with Mount Sinai School of Medicine. So we produced the antigen, which was the extracellular domain of ALK at LifeArc, and then it was a standard mice immunization, but we had two different groups where we did recombinant protein or use the DNA just to, so the mice would make the protein themselves in the right formation. After the immunization, made hybridomers from these, and this was Mount Sinai that did this part. And then we ended up with 1,152 clones. They did some initial screening of this using ELISA and flow cytometry to look for binding. And they looked at mouse ALK protein, human ALK protein, and then cells overexpressing human ALK. And from these various different readouts, we then took forward 53 ALK positive binding clones. So I'm going to talk through now the cell-based assays that we set up in-house to screen some of these ALK hybridomers. So it started with needing to look for target expression and antibody binding. So to do this, we used flow cytometry using the IntelliSite IQ screener. Next, we needed to check that there's internalization of these antibodies, so there'll be good ADC candidates. And we did this using flow cytometry and also imaging using the IncuSite Zoom and S3. And then finally, we needed to show that there's cytotoxicity. So we used a cell viability assay, set this up with toxin conjugated antibodies, and again used the IncuSite Zoom or S3 for this assay readout. So I'm going to take you through the various different screening methodologies we set up, and then also further candidate antibody characterization using these different methodologies. So first of all, for target expression and antibody binding. So often in papers, as we've seen for ALK, there's different levels of target validation as to whether the target is expressed in cells, and then also we need to look at whether the target is expressed on the surface. It's very important for ADC therapies that they're expressed on the surface, and there can be different readouts. Often in papers there's just qPCR analysis, whereas we need to look at the protein expression and also on the surface of the cells. So we looked at this for neuroblastoma, and I looked taking the initial bleeds from the mice. We looked at the protein mouse sera and those immunized with the DNA mouse sera. So this will be polyclonal sera, but it just was to guide us with cell line analysis to see which was going to be the best target for neuroblastoma cell lines. So we've got our range of whole different cell lines in. And using flow cytometry in grey, we've got the isotype control or the normal mouse sera protein to screen with as the background, and then compared those immunised with the DNA and those immunised with the protein. And as you can see, the vast majority of cell lines didn't show surface ALK expression, even though they're reported to express ALK. But because we want to look for this ADC approach where we bind to the surface of the cells, we needed to have surface expression. However, we did see intermediate expression with the IMR32 cell line. And as you can see with the mouse immunized with protein, there's a greater expression, so there's a greater antibody production in these mice compared to those with DNA, because they have to first produce the ALK protein and then produce the antibodies to this. And also MB1 cell lines, so we had two different cell lines, IFO and JCRB, and these both showed really high ALK expression. And this correlates with ALK overexpression, which is known in the literature. SKNES was a wild type ALK expressing cell line, but not on the surface. And there were various cell lines with some of these mutations, but these hardly showed any surface expression. So if I look at this in histogram format using the 1 in 1000 serodilution, you can quite clearly see that the vast majority of cell lines didn't show any ALK surface expression, but these MB1 IFO cell lines were showing good ALK surface expression, and IMR32 was showing an intermediate ALK surface expression. So this correlated pretty much actually with total ALK expression. So when we did Western blotting of these different cell lines, the highest expression levels were shown in these MB1 cell lines, intermediate with IMR32s. There was some intermediate expression shown with some other cell lines, but this wasn't shown to be on the surface, so it's not useful for us. And also look by imaging. And as we can see, so we use the in-cell analyzer high throughput cell content imager. So SKNES doesn't show any surface expression. IMR32 shows some cells, you can see the green rings of expression on these cells, but the MB1 IFO cell line was showing pretty much all the cells with nice rings of surface expression of ALK. There was this other cell line JCRB, and this shows equally nice surface staining expression, but as you can see, these cells don't grow as well, and they often form these debris. So we decided to focus on these three different cell lines. So we've got MB1 IFO that had the high surface expression, IMR32 with intermediate, and SKNES with no surface expression. So we took these three different neuroblastoma cell lines to screen the 53 hybridoma supernatants that we'd got from the ALK immunization campaign. So we set up using the IntelliSite IQ screener, and as you can see from the plate layout here, we've got the unstained cell line, and then we're looking at the normal mouse sera, and then the DNA and the protein mouse sera, and then all the different isotype controls. Then we've got the various different hybridoma supernatants in each different well. And then we use the different cell lines as well in here. So we've got SKNES, we've got the IMR32s, and we've got the MB1 IFO cell line. And we did this in 96 well plate format, first of all. So the good thing about the IntelliSite IQ screener, it's really high throughput and fast, and also the software to visualize really easy to see. So here we've got the histograms from each of the different cell lines. For SKNES here, we've got the ALK surface negative or very low, and here's the controls. So we've got the unstained and the isotype, or the normal mouse sera in grey and black, and then we've got the DNA mouse serum in orange, and then the protein mouse serum in blue. So there's an ever so slight shift with this DNA mouse serum. And then with the different hybridoma supernatants, you can see there's hardly any shift seen, showing that none of our hybridoma supernatants are binding to this cell line as expected. We can also look at the medium fluorescence intensity, and then here we get the readout of that in this readout here. So you can see quite obviously with the heat map which ones are showing strong binding. So the strongest is with the DNA mouse serum, but this is such a small, it's normalized to that. And then if we look the same thing with IMR32, so this is intermediate ALK surface expression. Now we're seeing the positive control, the protein mouse sera, showing a nice shift, DNA mouse sera showing an intermediate. And then with these hybridoma supernatants, you can clearly see there's some examples where they are showing an increased shift. And again, with the heat map, you can quite clearly see with the colours and the medium fluorescence intensity, which ones are lighting up quite easily. And finally, we did this with MB1 cell lines. So again, this was giving the highest ALK surface expression, and the serum from the mice immunised with protein are showing a really strong shift here. And then again, we can see with the hybridoma supinatants, there's some good shifts shown there as well. And we can easily see with the heat map that this hybridoma supinatant here is giving a nice shift. So I've converted this into histograms. So I've converted it from the different hybridoma supinatants compared to the isotype control, because we isotyped all of them. And then we can see that the main shifts here, which are giving over 10-fold shift, is the protein mouse sera on the intermediate and high ALK surface expression cell lines. And then there's a couple of the hybridoma supinatants which are showing up, but generally we're getting much lower shifts on these neuroblastoma cell lines. So we decided to repeat the screening just to make it clearer and give a big signal window to look at surface binding using overexpressed cell lines. So we looked at XP293 cell lines and overexpressed ALK in them. So it's a similar format where we're screening this hybridoma supinatants. We've got unstained, we've got the normal mouse serum protein and the DNA mouse serum, and then the different isotype controls as before. And then we've got all the different hybridoma supinatant candidates that we're looking to screen for surface binding. So this time we're looking at overexpressed cell line with human ALK, with mouse ALK, and then untransfected cell line. So similar setup to before. So this time you can see that the mock transfected cell line didn't show any surface expression of ALK as expected, and the mouse and the human ALK are showing good strong shifts with the serum from the immunized mice with the ALK protein, and then an intermediate shift, those immunized with the DNA. And again, we can see here quite clearly that the hybridoma supinatants are showing nice shifts binding to the human and the mouse overexpressed cell line, but not to the mock transfected. And again, if we look at the heat map of this, we can quite clearly see there's different colours coming out. So the strong binders are the more red colours, and blue is for the ones that aren't binding. And if I convert this to histograms, it's easier to see compared to the isotype control. Now we're getting a much better range, so we can quantify which ones are binding better or not. And we took anything above 4-fold shift from the isotype control, so showing a good amount of binding to the surface. So we ended up with 20 ALK positive binding clones from these 53 hybridoma supinatants. So next we wanted to look at internalisation. Now we've got some candidate hybridomas that bind to the surface and bind to ALK. So we used flow cytometry or imaging for this methodology that we'd set up. So first of all, I tested this method called on-bead conjugation. So with the murine isotype controls and the 20 hybridoma supinatants that were shortlisted from the cell binding, you can bind to protein A or G beads using the hybridoma supinatant. So therefore it binds just the Fc portion of the hybridomas, so the antibodies, and then you're able to add your reactive dye, small molecule, which can then be conjugated to your antibodies, and then you can wash and elute, and then you've got your labeled antibodies that are then purified from all the other proteins and cell sera from the hybridoma supinatants. So we tried out this methodology using the isotype controls and the 20 hybridoma supinatants we're taking forward. And here I've got the gels that we've got. So this is the unreduced gels as an example, and this is looking in fluorescent mode, so looking at Alexaflor 488, which we conjugated to them. And you can see with the Kumasi staining as well that we're getting good recoveries of these different hybridoma supinatants, and we're getting good fluorescent conjugation, so the dye is conjugated well. And then if we reduce them, we can see that the dye is conjugated to both the heavy and the light chain. So we use this acid wash methodology first of all. So you bind your conjugated hybridoma, so your antibody, to the cells overexpressing ALK, allow them to bind at 4 degrees C, and then wash off any unbound antibodies. Then we move the cells to 37 degrees C for a time course to look for any internalization of the ALK with the antibody, and then split this cell sample into two. So we've got one sample where we washed with PBS, so you've got the total amount of antibody bound, so you've got the surface and the internalized. And then we washed with acidic glycine, a pH 2.4, and then this strips the surface antibody, and then you're just left with the internalized antibody to read out. So an example of this, so at time 0, so in black we've got the unstained, in grey we've got the isotype control, and then we've got the two different samples. So at time 0 here we've got the PBS sample, so this gives the total binding of the surface and any internalized, but because it's time 0 there won't be any internalized. So this gives us the total binding signal. And then we use this acid wash at low pH, and you can see we're able to strip all the surface binding pretty much back to the isotype control levels. So the assay looks like it's going to work. We can get the total binding and we can strip with the acid wash. And with the mock transfected cells, we don't see any binding, which is as expected. Then after an hour, leaving the samples at 37 degrees C, we repeated the same. So we get the total binding in this PBS wash sample, which is pretty much similar to the time 0, because we've got the total at the surface and internalized. And now we've got the acid wash sample, which is now showing a shift compared to the time 0. So this is the readout of the internalized hybridoma supernatans. And if you look at a sample that's been kept at 4 degrees C for an hour, this is another positive control. You can see the total binding still the same, but when you acid wash, we can strip all the surface binding back to the low levels. And then we're able to quantify the amount of internalization. So after 1 hour at 4 degrees C, using the MFI values from the acid wash sample minus the isotype control, and then the PBS wash sample minus the isotype control, we're able to see that we don't get any internalization when the cells are kept at 4 degrees C as expected, but when we incubate the cells at 37 degrees C, we're then getting 31% of the total antibody that's bound internalizing. So we did this with all the 20 binding hybridoma supernatants, and we ended up with 12 internalizing candidates. And then we sequenced all of these just to see if there was differences in the antibodies, and then we ended up with 4 related sequencing groups. And from this, we ended up with 2 lead candidates that we took forward for further characterization. So in the meantime, when we've got these candidates, we actually came up with a better strategy because the cells didn't like being stripped in this low acidic glycine wash, low pH. We're able to use this tweaked methodology. So it's similar. We're able to bind the conjugated antibodies to the surface, wash away any unbound, then leave them to internalize as before at 37 degrees C. Wash in PBS to give the total, so that's the surface and the internalized antibody. And then instead of acid wash, we can incubate with anti-alexafluorofluorate antibody, which then quenches the surface fluorescence, and then we just get a readout of the fluorescence from the internalized antibody. So I can give you an example here. So again, we've got unstained in black and the isotype control in grey. And this is after 1 hour at 37 degrees C. You can see the total signal is in green here, which is the PBS wash. So we've got the surface and the internalized antibody. And then with the quench sample, you can see we're just getting a readout of the internalized. So there's a smaller shift. And if we do this at 4 degrees C, you can see that we're able to quench fully the surface expression, because if they're incubated at 4 degrees C, there's no internalization, but we're getting the same total signal. So plotted this over time and looked at some different samples where we've got the PBS wash, which is the total signal, and we normalized it to this over time, and then the quench samples to give a readout of the fluorescence inside the cell. So we looked at various different time points of this candidate hybridoma supernatant, the candidate antibody binding to ALK, and you can see there's a steady increase in internalization over this 3-hour time course. And we reached about 50% internalization after 3 hours. Another way that we can look at internalization is to look at imaging. So you can get these pH sensitive dyes and conjugate this to your antibody, and then as they become internalized into a more acidic environment of the endosomes and lysosomes, they fluoresce more at this low pH. So it's a good marker. It won't fluoresce when it's at the normal pH of the cell, so when it's binding to the surface. It's only when it's internalized that it will give the fluorescent signal. So you can see here the imaging we did on the IncuSite Zoom with the isotype control, and you can see over time that there's no red fluorescence because the isotype control is not able to bind. Whereas with our candidate antibody ALK1 on the HEC overexpressed ALK cell line, you can see there's some red fluorescence starts appearing at 2 hours, and then this steadily increases till 24 hours. And if we quantify the amount of red fluorescence, and then I've normalized this to the confluency of the cell, so we take into account cell growth, then you can get a normalized red confluency out, and you can see the isotype control isn't internalized, whereas the candidate antibody ALK1 conjugated to this pH rhodo-red dye is steadily increasing in internalization till 24 hours. And this time course takes a bit longer than the flow readout because it takes longer before you get the signal, because it has to go into the acidic compartments. So that's the discrepancy between the two different assays. Also looked at high content imaging just to confirm what we were seeing, because you can get much higher magnification on the in-cell. So we looked again at the HEC 293 overexpressing ALK cell line. So here we've got the candidate antibody conjugated to Alexaflor 488. So it's a green fluorescent signal, and you can see here at time 0 we've got a nice surface labeling of this cell line, so nice rings around the surface of the cells. We used a lyso tracker red dye to mark the lysosome so we could co-localize when it's internalized, and in blue we've got Hirsch for the DNA, and we've got the bright field readout as well. So over time, with 3 hours, you can start to see punctate green dots as the antibodies internalize with ALK, and at 6 hours even more, and 24 hours even more. And this, as you can see with the overlay, is you get this nice yellow overlay when it's co-localizing with the lyso tracker red dye. So we can see that we are getting internalization to the lysosomes. And with HEC 293 wild type, so no ALK express, we're not getting any binding at all, as expected. So now there are reagents available instead of having to conjugate your antibody directly. So since we've set up all this screening methodology, Sartorius have brought out the different reagents. So it's called FabFlor. So you're able to get a Fab that's conjugated to the pH sensitive dye. You can incubate that with your antibody to label it, and then look at the internalization. So it's similar, it becomes more red fluorescent as it becomes internalized to the more acidic compartments. So you can use this on the IncuSite, so look at the red fluorescence imaging of the cells as they internalize your target antibody, and also on the IntelliSite IQ screener to look at the internalization of antibodies and screen much more samples. So finally, after we've looked at the internalization of our candidates, we wanted to check that these antibodies, once they're internalized, are able to kill the cancer cells. So we looked at cell viability with the toxin conjugated antibodies, and again used the IncuSite Zoom or S3 for this readout. So we used Moradec secondary ADC technology. So instead of conjugating each different candidate antibody to a cytotoxic payload and then screening all of these primary antibodies, because it's expensive and time consuming, you can buy these secondary ADCs. So a bit like the FabFlor reagent, where the different secondaries you can buy, so they're either different anti-species. They're either full-length IgGs or Fab fragments, and they've got the different cleavable or non-cleavable linkers and various different cytotoxic drugs you can use. So as in the clinical samples, we took forward some of the secondary ADCs to test on our system. So I set this up first of all using HER2, so trastuzumab, as the positive control, and Kadcyla is one of the main, the first ADCs to be in the clinic that has been kept there for breast cancer. So I looked at a range of different cell lines. So we've got MDA MB231s, MCF7s, which are both HER2 negative or low expression, and then SKBR3, which is HER2 positive and overexpressed, and BT474s. So we used the IncuSite first of all to set up the correct cell seeding density. So I plated various different from high to low seeding densities of each of the different cell lines, and then looked over time at the proliferation of these cell lines, and you can see quite clearly that these are too high density, and then we're getting nice proliferation of this cell density, and then not enough cells at this lower level density. And we can get the nice 384 well overview looking at the microplate view using the software. And also can cope very easily with the different morphology of the cell lines. So as you can see, we've got very different morphologies of the cell lines, but the IncuSite is able to mask these very clearly and give the correct proliferation and confluence readouts of them. So if we look at the different replicates and plot them all together, you can see that over time, the lower cell density isn't proliferating enough, and the higher cell density is too confluent to start with. So what we're looking for is leave the cells overnight, and this would be the time point when we add the toxin or the ADC. So we want to start at low cell density and get to a higher cell density so that we can then see effects of the ADC. And for each cell line, we then optimized this. So you can see it's a different cell density for each different cell line. So we did a multiplex cell viability assay using parameter cell titer glow as a readout of how viable the cells are, and then cell tox screen assay for cell size toxicity. So we seeded the optimized density of cells that we'd found in the previous slide in a 384 well plate and left them overnight to adhere. And the next day we added on our control toxins, staurosporin and cell tox screen, or looked at the primary antibody in a concentration response curve, and then added on consistent amount of the secondary ADC. So we used FC anti-human with a non-cleavable linker with MMAE, or with a cleavable linker and DM1. So these are the two ADCs that were found in the clinic at the time when we started setting up this assay, and added on the cell tox screen as well. So we incubated the cells for 3 days at 37 degrees C in the IncuSite Zoom, and looked at phase and group images. Added on the cell titer glow assay reagent for an endpoint read at 72 hours, and read out the luminescence and fluorescence on a cell plate reader. So the results here are from HER2 negative cell lines. So we've got here the MB231s. So we've got here the viability. So as you can see with all the different treatments, it was just the staurosporin that was causing a loss of cell viability as expected. So the isotype control with the toxin isn't able to bind and cause cytotoxicity, and trastuzumab isn't able to bind to this cell line. The cytotoxicity readout was a bit more messy, a bit more variable, but again, you can kind of see the reverse. So there's no cytotoxicity with the majority of it, and then just staurosporin causing this increased cytotoxicity. Similar results with MCF7 because these are also HER2 negative, so the antibody trastuzumab isn't able to bind. With the HER2 positive cell lines, though, we are able to see staurosporin able to reduce the viability of BT474 and increase the cytotoxicity, even though this readout was a lot more messy to quantify. SKBR3 gave the best results. We were able to see this reduction in viability and then the corresponding increase in cytotoxicity, looking at the the readout of that. And then now we're able to see trastuzumab with the different secondaries is able to bind and cause internalization and then cytotoxicity, and the isotype control with this secondary FC ADC isn't able to cause that effect. So the assay looks like it works. We also read it out using the IncuCyte zoom rather than the endpoint read. So this is a nice plate map just to show you what's going on. So this is with the two different HER2 negative cell lines. Then we've got our 100% control, the lysis control. And then we've got our curve of staurosporin as a positive control. We've got our isotype control and trastuzumab going from high concentration to low concentration. And then we're adding on a constant amount of the different secondary ADCs from Moradec. So we're able to get the confluence readout and the green object readout cytotoxicity now on a kinetic readout rather than just the endpoint at 72 hours. So you can see the lysis control here is showing reduction in viability and an increase in cytotoxicity, and the highest concentration of staurosporin showing a reduction in the confluence and an increase in the cytotoxicity as expected. We're just getting normal proliferation of the cells with the antibodies because they're not able to bind. So if then we put these all together, we can look at the kinetic readout now with MBA and MB231s. So we've got the untreated, so this is the normal cell proliferation. And then you can see over time that the staurosporin is having an effect on the reducing the confluence of the cells and then increasing the cytotoxicity readout. And the antibody, so trastuzumab with the secondary ADC, isn't causing any of this effect. So if we take the area under the curve, we can then get a nice readout as before, like with the endpoint assay. And now we can see we get much cleaner results. So we've got the reduction in the confluence with staurosporin and an increase in the cytotoxicity. And now this is comparable to what we were seeing with the cell titer glow readout, apart from the cytotoxicity, which was giving us a pretty messy result. With the endpoint read, is now giving a much cleaner result using the IncuCyte zoom. Also did this with the HER2 positive cell lines. So now we can see with SKBR3, we're getting this reduction in viability with staurosporin, our positive control, but also now with trastuzumab and the ADC showing this cytotoxic effect, which we can see the increase in cytotoxicity here. And this is very comparable now to the endpoint read using cell titer glow or cell tox screen using the fluorescent plate reader. But the benefit of using the IncuCyte is that you get this kinetic readout data. So you can see at what point the toxin is starting to internalize and have its effect. So staurosporin and also trastuzumab, where you can see it takes about 24 hours to internalize and then start this cytotoxic or reduction in the confluence and increase in cytotoxicity. And this is consistent, that it takes about 24 hours for the trastuzumab to be internalized. So now we're getting a really nice kinetic readout, and you can see the nuances of different candidate antibodies now to see the timings of when they're starting to internalize and cause this cytotoxicity. So we now looked using this methodology with the primary ADC on neuroblastoma cell lines, so for the ALK target. So we looked at ALK1 and ALK2 conjugated to a toxic payload, and we directly conjugated these instead of using the Moradec system now, because we had the candidate antibodies. We went at the screening stage. So we wanted to check that they were able to internalize and cause the cell cytotoxicity. So we outsourced to ADC Bio the conjugation to a highly cytotoxic payload. We looked at an endpoint read using the cell titer glow readout, and then for the various different cell lines, so SK and NS, you can see there's no ALK on the surface, so we're not able to get a cytotoxic readout. IMR32 had the intermediate ALK expression, and we are seeing pretty much the same toxicity with both candidate antibodies. With HEX overexpressing ALK, we're seeing both candidate antibodies were able to cause this cytotoxicity with pretty similar IC50s. However, for the MB1 cell line, which overexpresses ALK on its surface, we're only getting chimeric ALK2 able to cause this cytotoxic readout, and chimeric ALK1 wasn't. When we went back to look at the binding of these candidate antibodies using flow cytometry, we can see that this is explained by the binding of the different candidate antibodies. So on ALK overexpressed cell line, we're getting good strong binding of ALK1 and ALK2. We're not seeing any binding to the surface of SK-NAS as we'd expect. With IMR32s, ALK1 binds much more strongly to IMR32 than ALK2, but they do show the same cytotoxic readout. However, the interesting one was that ALK1 isn't able to bind to the MB1 cell line, so this is why we're only seeing cytotoxic readout with chimeric ALK2. And if we looked into the literature, we're able to see that wild-type ALK, we think our candidate antibody is binding to this first MAM domain because MB1s have got a deletion in this exon coding for the MAM domain, whereas chimeric ALK2 is able to bind to MB1 cell line. So this explains that we were getting different results with our two different candidate antibodies. So we also looked at the kinetics, therefore, on the incucyte zoom of the ADCs internalising on the neuroblastoma cell line. So we compared ALK1 and ALK2 looking on the IMR32 cell line. So we're able to see the cells proliferating first of all, and then after about 20 hours, which corresponds to the imaging we were seeing on the incucyte zoom, we can see this internalisation and then the start of the cytotoxic readout. So we've got the different concentrations here. We've got high to low concentrations of the different candidate antibodies. And if we take the readout at 72 hours, which then corresponds to the point where we're looking at the end point with the cell titer-glow readout, you can see this corresponds. So we've got the high concentration pretty much causing the maximum reduction in viability, and then this staggers down like we're seeing on this CRC for the different toxins. So we can get this kinetic readout and start seeing the nuances of when the different concentrations are taking their effect and having the cytotoxic effect on the cells. So to summarise, we've got different cell-based assays we've set up. So we've got the target expression and antibody binding to make sure your cells express the target or that your antibodies are able to bind. So we used flow cytometry with the IQ screener. Next, we want to see internalisation. So we use flow cytometry methodology for this to look at the internalised screening of the candidates, and also imaging using the incucyte zoom or S3, where you see the red fluorescence with the pH sensitive dye. And finally, we looked at cytotoxicity. So we used either directly toxin conjugated antibodies or using the Morodex system, where you can put a secondary ADC on, and you get to see with the incucyte zoom or S3 the kinetic readout of this. So that just leaves me to thank the team at LifeArc who were involved on this ALK ADC project, and our collaborators in Mount Sinai School of Medicine who helped us with the immunisations and the hybridoma production. I just wanted to leave you with this flow of how we've integrated both the incucyte and the IQ screener. So you can use them for assay development, screening, lead profiling and lead characterisation, where the incucyte gives you the kinetic image-based advantages, but the IntelliSite gives you the speed and the high content. And we've used this in various different stages. So for assay development, so we set up for the cytotoxic readout, we used to set up the cell densities to plate. For screening, we used the IQ for screening the hybridoma supernatans binding to the cell lines. And then lead profiling, and we used the IntelliSite IQ screener then for internalisation of all the different candidates. And we also used the incucyte zoom or S3 to then image this as well. And finally, for lead characterisation, we looked at the kinetics then of this internalisation and cytotoxic readout. Thank you very much.
Yeah, thank you so much for your time, and